Sarah Watts, who's going to talk to us about mycorrhizal associations, microsite factors, and natural regeneration potential in montane scrub restoration.
So yeah, it's been really great to be invited along this evening to talk to you all about my ongoing PhD project, and nice to hear about Annabelle's work just then as well, kind of following on the theme of PhD research, which is really nice. So my PhD project's called Improving Outcomes in Montane Woodland Restoration, based at the University of Stirling. And I'm very much in the midst of my PhD, so the talk is really to kind of give you an overview of what I'm currently doing rather than discuss kind of a whole lot of results. But hopefully I can perhaps come back another time or give a future update on what my research has found when the project's finished. So there's plenty of photographs to show you today of what I've been up to as I'm kind of running through the 4 main research questions or chapters of my project. So firstly, to introduce myself, I did a degree in ecological sciences at University of Edinburgh, graduated in 2012. Then I worked for 8 years as the seasonal ecologist for the National Trust for Scotland at Ben Lawes NNR, which was an amazing opportunity to monitor really exceptionally rare special habitats and species of conservation interest in an upland environment. And that's really how I got into upland botany. Upland ecology is my specialism. And the work also involved monitoring outputs of restoration work ongoing at the site. But as well as loving the fieldwork itself, I also found myself getting really excited about data analysis, experimental design, and writing about my results. So I kind of thought it would be really exciting to explore that avenue further and pursue a PhD. And as Ben Lawes is a site pioneering mountain woodland restoration in Britain, it really provided the inspiration for developing ideas for the PhD project. But then fast forward further on, and now I'm through 2020, and now I have a job as a conservation manager at Carrowa Estate, where I've been working part-time for the last 1.5 years. And in addition to that, also taken over the role of chair of the Mountain Woodland Action Group, formerly Montane Scrub Action Group, previously chaired by Dana Gilbert, which is really exciting opportunity. So as I said, the PhD project is called Improving Outcomes in Montane Woodland Restoration. And this is a part-time project, so it's 6 years in total rather than kind of usually 3 or 4. And I started in October 2019, so due to finish in 2025. So I'm coming up to halfway through at the moment. And the project's one that I devised myself rather than applying like for an advertised project, but that also meant that I had to find the funding for it myself, which I managed to do, fortunately, through a consortium of 8 different funders who I'm exceptionally grateful for, making my dream research project a reality. So the aim of the project is to aid the development of conservation management techniques to restore healthy and sustainable populations of montane trees. And I really developed the idea kind of in conjunction with ecologists, conservation and land managers working in or hoping to start mountain woodland restoration projects, asking them what they thought would be most useful to know in a research context for helping to inform the direction of their habitat restoration work. And so the project's very broad. I'm coming at it from several angles, kind of looking to the past, present and future timescales. So looking at pioneering restoration work to understand how we might build on that knowledge for informing new work that's getting started now. And then also trialing some new restoration techniques in these new projects. And then finally looking to the future and thinking about, creating self-sustaining populations of montane trees through encouraging natural regeneration. And all of this been done through a combination of field observations of montane trees in situ and also experimental field trials as well. So it's designed to be really very applied, outcome focused, to deliver outputs that will be relevant and useful in the real world when people are designing their own habitat restoration projects. Hopefully there'll be lots of information we can disseminate through organizations such as yourselves and the Mountain Woodland Action Group, as I said, just taken over a role as chair of that group. And the focus of the projects primarily on montane willow scrub, particularly downy willow, just because of the experience I've had at Ben Lawes, but there's also an element looking at dwarf birch as well. So to explain why we want to restore mountain woodland and scrub in Britain, I'm sure you're all very much familiar with this premise, but nevertheless, there has been a significant habitat loss of mountain woodland and scrub in Britain. And what we have now is very much a remnant of a formerly much more widespread habitat type that has been reduced over the last few centuries due particularly to overgrazing by sheep and deer. So we've really almost got a complete lack of the altitudinal tree line ecotone in Britain, which means we're missing this transition zone between where trees have a very upright growth form and then moving up towards more stunted sort of prostrate trees and shrubs all the way up to higher altitude alpine grasslands and heaths. But there's more to restoring this habitat than just because, you know, it's nice to do. Reinstating mountain woodland has significant benefits for biodiversity, ecosystem services and nature-based solutions, particularly for mitigating impact of climate change. So thinking about encouraging slope stabilization, protection from soil erosion. They also reduce downstream flooding risk and provide sheltering. And this is such a key topic that I've recently written a short mini review paper on the benefits of mountain woodland restoration, published in the journal Restoration Ecology, which is designed, well, it's completely open access, so anyone can read it, and it's designed to be read by a wide variety of people, you know, land managers, conservation managers, ecologists, and the media general public, just to kind of get people inspired about why this is such a good thing to do. But moving on now to think about the actual chapters of my PhD, with that review paper kind of being part of the introduction, so the chapters, the data chapters of my actual research. So the first chapter is focusing on established planted montane scrub habitat to try and understand where the biggest, most productive shrubs are growing. And kind of translating it as a mechanism for developing our knowledge of where we could plant seedlings for good outputs, good growth outputs in new restoration projects, and also where we might try and avoid planting if we want our shrubs to survive and thrive for decades to come. And it just so happens that one land manager who'll be implementing a new montane scrub restoration project in the near future is actually me, through my new job as conservation manager at Karawa. So that provides huge motivation to deliver high quality research, because I'm myself going to be one of the people using the outputs. So the study site for this part of the PhD is Ben Lawes NNR. Of course, the amazing population of downy willow, Salix laponum, which was planted at Ben Lawes about 18 years ago, and completely pioneering. There's no other population in Britain, planted population of this scale or age of shrubs, so it's completely unique, although of course there are now other sites following their lead, which is super exciting. But what I found was really interesting about this site when I got to know it through my work is that there is quite a huge amount of variability in the growth of the shrubs there. So some of them are really, really big, like, you know, nearly 2 meters tall. And then others, it's the same age, but really, really tiny, stunted, you know, they might be only 30 centimeters tall. So I was thinking, well, what's driving this variability in shrub size, even though they're all planted at the same size? And this site is about 600 to 750 meters in altitude, all within a fence to keep out the large herbivores. So just a bit about my data collection. This was all taking place last summer in 2021. So I had quite a task ahead of me, measuring loads of different growth attributes of the planted Salix Aponum at Ben Lawes. So shrub height, canopy dimensions, canopy density, stem diameter, and interannual growth, using bud scars on stems to kind of track back previous seasons' growth, which I find a really interesting measurement to make. And then also collecting a whole load of sort of site variables, abiotic and biotic site variables at the level of the individual shrub. So all of these measurements were made at each individual shrub that I measured. So think about altitude, slope, angle, aspect, soil depth, vegetation height, just some of them. And then also recording the sort of cover of various different species groups of associated plants growing with the willows, and then also percent cover of the most dominant species growing with the willows, to then calculate Ellenberg site indicator values for each individual shrub. So a huge amount of measuring went on last summer. So bearing in mind that this is only part-time, I'm working on this part-time in addition to my job. So I had 31 field days at Ben Lawes on this work. I managed to sample 1,050 individual bushes, giving me over 55,000 individual measurements. And for a data set of which I am incredibly proud of, and still very much in the process of analysing, because I'm part-time, so these things take a bit longer, and I'm learning techniques as I go in data analysis, which is super exciting. So I just thought I'd share you a few quick kind of highlights as a kind of idea of what some of the things the data are starting to show in terms of response of the shrubs to different site characteristics. So these charts don't need to worry too much about what growth PCA means. Basically, all my growth variables, the height, canopy area, stem diameter, etc., they were all correlated. So rather than do them kind of in models, data analysis individually, which would be repetition, you can do a process of ordination, which basically combines them into like one super variable, which captures everything together, called a principal component analysis. So that's what the growth means, but high values are big shrubs and low values are small shrubs. So the variables at the top, particularly fertility, vegetation height, soil depth, they all had a positive effect on growth. So bigger shrubs were growing in areas of high soil fertility, where the vegetation, the surrounding vegetation was taller and the soil was deeper. And then at the other end of the spectrum, we have smaller shrubs growing in areas of wet soil, where there's a high cover of sedges and rushes as well. So I was really excited to see such huge responses in my data to explain the variability in growth. But it's not necessarily all about growth, because at the end of the day, you might want to think about reproduction as well, because maybe huge shrubs actually aren't producing any seed, and then that's not really very useful for long-term sustainability of the population. So amongst other things that I'm currently analysing at the moment, I'm also looking at what might affect the probability of shrubs producing catkins. So this shows probability distribution, probability of shrubs having catkins, which the main thing to focus on here is that there's a huge decline in the probability of shrubs having catkins as the cover of sedges and rushes increases at the site. So the kind of take-home message is that if you plant in areas of high sedge rush cover, you'll not only have very small shrubs, but you'll also have small shrubs that aren't producing any catkins at all. So these are very much the places to avoid. So that's kind of a flavour of what I'm working on at the moment from the data that I collected last summer, which I'm finding really exciting to kind of tease apart what might help us direct future planting projects for downy willow. But the second chapter of the PhD is very much ongoing. This is, rather than making observations on planted shrubs in situ, this is actually a kind of experimental field trial that we're implementing specifically for the PhD, focusing on a wonderful species, dwarf birch, which I absolutely love working with. And the rationale behind this planting trial is that dwarf birch in Britain is kind of typically associated with blanket bog habitat, but there's kind of a recognition now through some ecological niche modelling analysis that's been done that perhaps it's actually adapted to a wider range of habitats, also looking at it growing elsewhere in Europe. And maybe it's restricted to blanket bog in Britain because of historical pressures of overgrazing and burning, rather than because that's necessarily its preferred habitat choice. So we wanted to see if the dwarf birch could grow equally well in different habitat types in contrast to blanket bog. So we chose blanket bog, sort of dry dwarf shrub heath, and upland grassland as well. So I've planted a whole load of dwarf birch in these 3 different habitats at 3 sites. So Ben Lawes, Carrower Estate, and the wonderful Glen Finglas with the Woodland Trust as well. So 300 trees at each site planted in these different habitats. And then in addition to that, there's also a grazing treatment as well. So half the trees are planted inside fences and half of them outside fences to see how they do with different levels of deer, with Carrower having a very low density of deer, and Glen Finglas is quite low as well in the Scottish context, to see if the deer graze the trees in the grassland and the heath more than the blanket bog, to help us understand what might be affecting growth and survival in addition to habitat type. So I'm very grateful for all the help that I've had with planting these trees. And the trees, well, the first lot went in at Carrower in 2020, and Glen Finglas went in last year, 2021. And well, Ben Lawes was before that, I think 2018. So what I'm doing is monitoring growth at the end of each growing season. So it's repeated measurements done on the same trees that have been planted at the end of each year, about October time, to see how they get on over time. And I've got several growing seasons throughout my PhD being part-time, which is useful for the slow growth rates in upland environments. And it's very much early days. I've only got a few seasons of growing measurements, so it's not that we're only just building up the picture of how they're getting on. But this is a kind of chart to just sort of show this kind of data that I will be producing. This is just after 1 growing season So the trees at Karawa were planted in 2020. So this is the data from 2021. So it's important not to draw too many conclusions from it because it's just one site after 1 year. But nevertheless, there was quite a strong result in the shoot length. So what this shows is that the trees planted in the heath habitat had a much shorter growth in that growing season in 2021. So they hadn't put on as much growth as the trees growing in the bog or the grassland habitats, which is very interesting. And at Karawa, it didn't really make any difference whether they were inside or outside the fence because very few trees had actually been browsed. So I'm going to be continuing with this work, really looking forward to getting out in October and seeing how they're all doing and hopefully building up a sort of long-term data set of, well, at least the initial years after trees have been planted in these 3 different habitat types. And then moving on to chapter 3. So this one is on mycorrhizal fungi, which is quite different from the rest of my PhD. But when I was asking everybody involved in mountain woodland what they thought might be most useful to research in this context, it was mycorrhizal fungi that everybody said. The rationale being that we know that it's important in general in woodlands, and that's becoming more and more apparent as research comes out, but very little is known about it in the context of montane scrub, montane woodland, and whether it's useful to consider that in habitat restoration projects. So the fact that everybody was saying that, well, I thought, well, I have to include some of this in my PhD because it's just so fascinating and who knows what we're going to find out. So it's still very much in the kind of developmental phase, actually, as kind of DNA sequencing and sampling technology develops. So I'm kind of riding the crest of that wave, which is really exciting. So kind of looking at this from 2 angles. Well, one thing that we're hoping to start this year is possibly doing a trial of inoculating planted downy willow with an inoculum that's being developed to see if that helps improve their growth and survival in the field. But in addition to that, I just am really curious to find out what's actually associated, what the actual fungal community is that's associated with these shrubs in the upland environment. Is it anything particularly unusual in comparison to willows in general? And if so, should we be doing something about it in our restoration project? So the first step is just to find out what's there. And a really exciting technology is being developed for sequencing eDNA, that's environmental DNA from the soil. So I've trialed this, and this is literally work that I did 2 days ago, collecting some tiny soil cores from a relict Salix saponum to send off to a lab to sequence to see what sort of fungi is growing there. Very much an exploratory analysis, but no one's done it before, so this is exciting, and I will hope to be doing this, upscale this work in September, October this year, collecting soil samples from 3 different populations in Scotland. So that's Ben Lawes, Carrow Estate, and also Mar Lodge as well, the Cairngorms, to just have a bit of a broader geographical distribution and see what we get. But it's very challenging work because if anybody has visited relic stands of downy willow, you will know that they're incredibly difficult to get to due to the fact that they're often only residing in locations that browsing animals can't reach, so that makes it difficult for people to reach as well. They're often on cliff ledges and completely inaccessible, so I'm kind of restricted in some respects to sampling to the trees that I can get to safely. And then even when you're there, you have to be very careful not to do any damage, because they're obviously growing in quite perilous locations and they're very old. So fortunately I have good knowledge of these sites, so that's helped me select very carefully which individuals I'm going to sample from, so that I don't do any damage to them or either myself as well. So finally, the final chapter of the project is looking at natural regeneration, which is so important because, of course, that really the long-term goal of the restoration work should be for natural regeneration to take over, so that the populations can be self-sustaining and we don't have to plant them anymore and they can just thrive and get on with it themselves. Obviously, with the recognition that for slow growing montane trees, that could take a very long time. But nevertheless, at Benlaw's, so the trees there, the downy willow had been planted about 18 years ago. And we were kind of concerned because we hadn't really seen any evidence of natural regeneration, at least when I was developing my PhD project ideas. So I was curious to sort of discover what might be limiting natural regeneration and if there's anything we can help to encourage it along. Partly with the worry that the population is within a fence, so it's producing this, where the exclusion of grazing has produced this wonderful ground flora, which is a conservation success story in its own right. But because willows particularly need bare or sparsely vegetated habitats for their seed to germinate, that actually the ground flora might be impeding natural regeneration in some way. And we were kind of hoping that there might be some regeneration in these active landslip sites at the restoration sites, so where soil is moving and creating bare ground niches. So kind of exploring various different reasons for what might be limiting natural regeneration, looking at the maturation of catkins on planted downy willow at Benlaw's, doing some seed viability tests in my greenhouse, and also looking at the availability of germination niches. But fortunately, last year I did actually observe for the first time seedlings of downy willow at Benlaw's. So this was actually a really neat opportunity to change the focus slightly and make the use of the opportunity of the seedlings that had just started to appear to find out where they're growing. So this is work that I'm going to do this summer, or have just started doing this summer. So taking observations of the seedlings in situ, looking at there's very specific micro habitats where they're growing. I mean, literally the immediate vicinity of each tiny seedlings, you know, which moss species they're growing on and where they are, to build a picture of what they need to germinate and establish. And then in conjunction of making observations of naturally regenerated seedlings in situ, I'm also trying to speed up my understanding of the process by doing some seed sowing experiments in the field as well at landslip sites, not landslips with the seedlings, so the other areas a bit away, you know, so that there's not confounding factors and seed blowing in and changing my experiments, but sowing seed in 4 different microhabitat types within landslip sites to see if there's any germination preference within these sites, to understand where the seedlings might grow, if there's enough seeds landing on the landslips. So this is work. I will be out on the hill doing this tomorrow and very regularly, twice a week for the next few weeks, and later on into the summer. So very much looking forward to seeing what my data is going to show from this field work at Benlaw's. So that is just a brief rundown of what I'm doing with my PhD, and yeah, it's incredibly exciting to be doing this project that I've sort of dreamed of doing for quite a few years. And I'm so grateful to all my funders in my funding consortium who've helped support it along the way. And very much grateful for input from land managers and conservationists, ecologists, everyone sort of involved in this kind of work and helping guide what would be the most useful research to help inform their work. So thank you very much for inviting me along to talk to you this evening, and yeah, look forward to take any questions.
Thank you, Sarah. Wow, that was amazing. Very, very interesting. Okay, does anyone get any questions for Sarah? One thing I was thinking about, Sarah, was just how vulnerable this habitat is. I mean, it's always occurred to me to be the sort of environment that it's almost ethereal. You know, it's something which you see now and again, but the scope and scale for the potential must be huge, which you're beginning to sort of find out about. How on earth, in a practical way, are we going to have any chance of protecting what we could restore? I mean, it must be, you know, an almost impossible task.
Yeah, yeah, because these populations, particularly of the rare montane willows, are so scattered and fragmented.
Yeah.
That some of them, you know, it's single individuals or a handful of individuals. So yeah, well, I mean, first of all, I guess you have got to combat the original reason for their decline in the first place, which is principally overgrazing. And that can be done through several ways. And I think it's not an either or choice between fencing out areas or landscape scale herbivore control, because, you know, it depends on the management of each individual site, which one of those options might be better. And there's advantages and disadvantages of both, but, you know, each does work to combat the problem. So that obviously needs to be done if you would like to expand populations. And then it's thinking about what, you know, using what they're doing at Mar Lodge currently as an example. So focusing on existing fragments of populations, but then doing supplementary planting to kind of join the dots to create a more continuous habitat across the landscape in suitable places. And that's what we're hoping to do at Karawa. So I've been out mapping all the existing populations of the downy willow, also whortle-leaved willow as well, which is Salix mercenites. And then there's very limited seed production. So we can collect a small amount of seed, but that in itself is not really enough. And then you've got issues with genetics if it's only coming from a few individuals. So we'll be collecting, well, we have collected cuttings from as many individuals as we can reach, and then putting them together to form a seed stand, a seed orchard. And then that can upscale seed production massively and mixing all the different genetics from the different populations across the estate. And then, then having those to plant out and join up the dots. So it's a very long-term project, and you have to be highly motivated and patient to wait several years to start seeing seedlings out on the hill, and then wait decades to see the next generation appearing. But having been inspired by Ben Laws in particular with what's happening there, I know what it will be like. So that's huge motivation to start work elsewhere. Okay,
thanks. Well, we'll all keep our fingers crossed. We've got just a few minutes left for a couple more questions. I see Ro has got a hand up, so we'll go to Ro first, and either Felicity or Andrew have got their hand up as well. So we'll just take those two questions, and I think it will be, you know, looking at the clock, it will be time to wind things up. So Ro, would you like to go first?
Yep, okay, this is a genetics related question. I was just wondering what's known, if anything, about the genetics of particularly the Salix leponum, and whether some plants naturally have a propensity to grow bigger than others, or as there's so few of them about, maybe we don't know much about it anyway.
Yeah, that's a good question, isn't it? And I have wondered about that, because you do get some trees that are just big shrubs, and in others that they do seem to just more be low, kind of sprawling. And you know, if they're producing lots of catkins and performing the function in the shrub scrub habitat, then that's fine. And actually, I would say, you know, this sort of structural variability is a good thing, structural heterogeneity in the landscape with sort of different sizes and dimensions of shrub, providing different niches for different birds and insects and things, rather than everything being uniform. So it's not like necessarily striving for always having really massive shrubs. It's more about the habitat in general. But yeah, I don't know if there's any genetic factor about that. I mean, there definitely must be. But the only genetic work that I know that's been done is by Aileen Finger in the RBGE in conjunction with Mar Lodge and Cairngorms Connect, taking leaf samples from different populations and seeing how related they are to each other, which helps kind of inform whether you collect your propagation material from a sort of smaller area to preserve that genetic identity, or whether you collect it from a larger area to include sort of improve genetic diversity. And I don't really know the answer to that. It's probably a bit of both.
Okay, thanks. Right.
Thanks, Ro. Thanks, Sarah. So final question to Felicity and Andrew, please.
Right, well, first of all, that's an incredible amount of work you're doing, and all that walking uphill must be keeping you fit. I just wonder how you precisely mark all these places that you're visiting. You've got so many different places you're visiting on all these hills. Are you using very accurate GPS to, know all these locations?
Yeah, so I do have handheld GPS, which is hugely valuable. So taking 10-figure grid references, so that for if I'm mapping relict natural stands on the hill of these species, then it's grid references and lots of photographs. You can never have too many photographs, and then annotate the photographs back at the desk. And then each individual gets its own unique identification code. So if I'm going back to get cuttings or anything, then I know who I'm collecting from. But then for the trees that are sort of involved in the growth measurement work, so thinking about the dwarf birch that we planted, they all get their own metal tree tag that's attached with wire at the base. So they've all got their own unique identification number. When I go back to measure them, I know which one I'm doing, and I've got them all mapped out, so I know where they all are in relation to each other in case any of them go missing, which is really helpful. And then the population at Ben Lawes, the planted shrubs that I measured last year, that was actually quite a challenge, because I had to do the sampling throughout the whole of the summer to get enough data, but some of the measurements could only be done like at one particular time of year. So thinking about vegetation height and leaf senescence and things, they would change throughout the summer. So they had to be done all together. So I sampled my 1,050 shrubs throughout the summer, and then had this week at the end of August where I went back and visited each one again a second time to do the second round of measurements. Which I managed to do in 4 days, despite the fact my supervisors thought it would be impossible to do it in 5. So I did it in 4 instead. And I basically had GPS'd each one and taken photographs. But actually the most useful thing was just drawing my own sort of hand-drawn maps of dots of shrubs, where they were in relation to each other. Maybe use the GPS in the photo to find the first one, and then just using the diagram to weave my way up and be sure I was doing the same individual. So yeah, you develop your own techniques over the years on how to find these things.
Did you camp out to cut down the time walking up and down hill?
No, well, I'm very lucky. So I live in Killin, which is very close to Ben Lawes, and the field site's only actually 15 minutes drive from my house. And then you walk across the Loch and the Larrig Dam, which is quite high at altitude already, 500 meters. And then you're basically about 15 minutes walk from the from the willows, so that was actually quite easy. Some of the other sites, well, Carrower and then particularly Mar Lodge, when I'm visiting the relic populations, there will be longer walks in, but having prior knowledge of these sites, you know what you're getting yourself in for. But you know, I don't camp out, certainly not for this particular work. Just long hill days and trying to be as efficient as possible, really.
Okay, thanks, Sarah. Thanks very much indeed. And as we said to Annabelle, you know, best of luck with everything. All the best for your project and your research. I'm sure you won't need any luck at all, but it all sounds like it's going very well and it's a fascinating subject. Thank you very much for giving us a benefit of what you found out so far.
Thanks very much for having me.